Most people here will know Professor Greg Snell, who's the medical head of our lung transplant service. He's been part of the service since 1992. He did his medical training at the Melbourne University and then went to do a Will Rogers Fellowship in Toronto and trained in lung transplant there, and has been basically back here at the Alfred ever since. Greg's an expert in all things to do with lung transplantation, particularly in research. He's published over 300 papers, 100 or so in the last 5 years, and I think that's enough for now. We'll move on.
Yeah. So thank you to everyone for coming along, and great to have the opportunity to talk. So I thought I'd sort of give an overview of the ripple effect of what we've done, and I was after collaboration, and friends with benefits seemed initially like a good idea, and then it started to occur to me that I was perhaps getting into a little bit of trouble with that. So I actually changed it to the ripple effect of the nerd herd. For those who, you'll hear Lucy and Steve talk later, and it's just a little surprising how that does seem to be modelled on them, actually. So, and with the help of Lungitude to do this, yes. So why are you guys here? Why are we talking about the Alfred Lung Transplant Program and our friends and collaborators? We are one of the world's largest, in the 5 largest programs in the world, and arguably one of the very most successful programs in the world. It's also true that Melbourne's research precinct, or NERDS, have a very huge research record and have contributed, and you could actually say that there are Melbourne institutes that actually invented immunology. Perhaps Lucy can clarify that a bit later. And end-stage lung disease are common. It's one of the commonest diseases affecting people, and there's an unmatched need for extra work and extra ability to fix things. We're in a position here that in Australia and in Melbourne we're able to think of things, work through them and translate them. Lungs turn out to be relatively easy to study. They're big, we can get into them and out of them with bronchoscopies and the like, and you get lots of cells and lots of tissue from our specimens. And indeed, the results that we find in these large organs can apply to little kidneys and other much easier organs to transplant. I've talked in the past to some of you about this idea where transplant starts with an ill patient and obviously an ill donor. The donors contributing their lungs. There's a waiting list. There's a lung transplant, and then there's your ultimate your quality of life and survival after a transplant. And we've essentially tackled this over the last many years. We've looked at different types of donors, the donation after brain death, donation after circulatory death. We've looked at infection, we've looked at rejection, and there are also lungs that are removed from the recipient with their end-stage disease that I'll touch on in a minute. So if you look at what we've been tackling over the last few years, first of all, we've looked at these donor lungs, and we've aimed to get as many lungs working as we can and from as many sources as possible. And there's been successful research that started many years ago and supported by the Margaret Pratt Foundation and Lungitude in more recent times, that has since flowed on through our DCD work and our extended donor work to be an Australian standard. The Canadians copied us. We did ex vivo lung perfusion before Shaft did. We did DCD, and the donation after cardiac death has been used mostly for ex vivo in Canada. The Belgians and others have worked with us, and for some reason I put Canada twice. But the Transplantation Society of Australia and New Zealand and the International Society of Heart and Lung now have these as essentially our protocols as their national and international guidelines, so we're very proud of that. If we look at the lungs that are removed, we've actually found out quite a lot about these donor lungs, and the idea that the lungs carry cells with them that then go through the circulation of the recipient, and it will I think be touched on by some of my other speakers today, but we've found that the transmission of cells can explain some of the rejection features that are seen in a recipient, and also some of the infection features that are seen. And from the lung work, we're explaining otherwise previously poorly understood mechanisms. And one of the challenges a little bit is, as we try and publish and talk, is to convince the infectious diseases people that these are passengers, not infections. They're just cells, not viruses and things. And it's the interesting phenomena of science of trying to change a paradigm with a new idea. And these rejection strategies are being worked through Steve and who you'll hear from later to try and understand some of the features of cross-matching. So we're very pleased with that as well. The lungs that are removed at the time of transplant have end-stage lung disease from a variety of conditions, and we've found that this has been a very useful resource for collaborators to be able to look at the lungs and try and understand the diseases that have cost that person their lung performance. And tissue specimens go to the Melbourne University and to Monash University. And we were I was particularly impressed about 18 months or so ago when we had some researchers from Monash, career researchers who'd been working on creating a new lifelong flu vaccine. And this lady had 3 sort of sections of lung from older patients, and she was dealing with real tissue that was preserved because it was surgically removed at the time of transplant. And she was just beside herself with excitement, describing how she was looking towards creating a single vaccine that you'd have for life with influenza by showing certain memory cells within the lung that was showing that the person had immunity from previous infections and things. And it was a really interesting moment as we just sat there to watch this lady just so excited after years of working in animal models. She's just thrown the animal stuff aside and said, I really must be working in the humans because this is really going somewhere. And treatments for pulmonary fibrosis and pathways for pulmonary fibrosis, and that the Melbourne University people are working on. We've also been looking at infection, and in particular, Lungitude supported us with looking at cytomegalovirus, which is a common infection after transplant of all types or in all immunosuppressed people, and trying to understand this balance between getting an infection but having the immune system strong enough to fight it off, but not so strong that it fights off the new transplant. And we've been working with a company here to finish the cytomegalovirus study, looking at a measure of how active the virus is and the immune system against it, and now getting the idea that we can measure the immune system and maybe get an idea of just how effective our immunosuppression drugs are. So using the effect on a virus to work out whether the drugs are too strong or not strong enough. So that's an ongoing work that the team's involved with. I touched on before involvement with the Red Cross Blood Service and how this has been a major project really that we're really looking to changing the whole way of matching. The Red Cross has created a new database system, a new matching system, and that we're getting in right at the ground level to be able to change the lung algorithm and get agreement to that around the country as to how to better match organs and how to be cleverer. And Steve will give you some examples of that later, what we're thinking of. In terms of avoiding rejection afterwards, we're trying to look at different types of tissue that you can use. Instead of doing our standard biopsies of the sponge of the lung, we're looking to biopsy the pipes of the lung and comparing that, and we're sending tissue off to Canada to have that analysed and look at and hopefully get signals from that are cleverer and more effective signals than we currently get with the tissue that goes off to our Alfred laboratory. There's also other tissues and other cells and other fluids that are being taken from the lungs that are being sent elsewhere and being worked on to try and understand the signals that create rejection. And Lucy will probably touch on the fact that we're even trying to look at ways of monitoring the cells that come out of the lungs and into the body and see which ones might predict rejection that before the person actually gets rejection. So this is proving very interesting as well. I mentioned they're the sort of where we've got up to in the past. If you're now sort of going forward, we're still got a big project looking at analysing the pattern of lungs that were used for transplant versus those that were thought unusable or not able to be used, working with Donate Life and the University of Okayama team. We have Japanese visitor in the audience. Thank you, Shuji, who's working on that. Trying to understand why we don't use organs at times, other ways we can improve the efficiency of the process. We've got a picture there of our machine of looking at the lungs on a rig. The lungs are tucked in the corner here. There's lungs on a machine that you can assess them, and there's a whole lot of pumps and stuff that run the fluids through and keep the lungs alive. And as I've mentioned, we also, down the bottom there's some pulmonary fibrosis lungs that have been chopped out, and they can be sliced up into bits and sent off to different laboratories to try and understand the pathways that got the person those damaged lungs in the first place. We've been looking at rejection, the matching. I've mentioned trying to improve that, working with the Red Cross and this new system of theirs called Organ Match. We're creating a national collaborative. We've even got an international collaborative, which we're slightly dubious about, but an international collaborative with one of our Lungitude-sponsored projects on rejection previously, who's now gone on to, the guy we worked with has taken the idea and is trying to run to the world with it. And Steve has a master plan to perhaps work with that, and we'll continue to try and improve a matching process. And Steve was just telling us before that various other sites in the US have signed up to have a worldwide information sharing thing. So we'll have to see how it works, but that's definitely come out of, this whole concept's come out of the work that Steve and Duncan before him had done. We've got trials of gamma globulin with CSL, randomised control trial there, which is interesting. We've got the cleverer diagnostics. I've mentioned the interlung study where stuff's being sent to Canada looking at the different fluids and different diagnostic ways. We've still got the Red Cross and the Doherty Institute and the Murdoch Institute looking at passenger cells from the lungs travelling around the body, and Lucy's working on very subtle measurements of DNA and being able to track even a fraction of the cells from the donor that might be in the recipient and might make you prone to rejection or not, and looking at those. Cleverer lung monitoring, that's a new breathing test technique, forced oscillatory technique of trying to work rather than the simple breathing test we do at the moment. Another treatment trial, another randomised control trial of intravenous stem cells to treat rejection when we've got it, which is we're part of an Australian collaborative with the NHMRC. Ongoing projects, we have the CMV quantiferon and quantiferon monitor looking at the immune system response to infection and whether we've got too much or too little on board. Another randomised control trial of an oral antifungal, part of an Australian collaborative that's being led by the team here at the Alfred. Part of that includes looking at fungus resistance and inflammation around fungus, the Aspergillus fungus. There's a microbiome study that Ben will tell you about in a minute. That's Ben's baby, and with the Monash University team. And Lucy will possibly tell you about her secret T-cell 11 herbs and spices anti-CMV recipe that she's looking to patent. The diagram in the right may look like a piece of artwork, but in fact Lucy sent it out as a clear example of a part of a publication of showing something. The figure legend just said 3 words or something, so I'd be interested to hear what it is, but I think it would actually look quite good on the lounge room wall, to be honest, Lucy. So that's very interesting work. We're actually, again, translating experimental work into hopefully a therapy that's patentable, fundable, etc., and I'm sure Lucy will tell you something about that. The actual translation of Alfred research compared to the USA, I think, is worth summarising here. In Victoria, we have, per million population, we have 31% less organ donors than they do for the same population in the US, so if we were an American state. Having said that, 69% less people will die on the lung transplant waiting list here in Victoria. 50% more transplants from the same population we're able to get, and each year afterwards, 57% less deaths with transplants. So these, and you look at 100 people and apply these numbers, and you're really starting to get very significant effects that make a difference to real people, not just, you don't have to do 1,000 transplants to see these effects. These are real, and that's summarised here where you've got essentially a survival curve taken from the time of transplant and the rest of the world, they'd lose 20 to 25%, 50% if you're in England on the waiting list, and we'd lose, unfortunately, still 4%, and then we're able to run better than the rest of them at right out of very long time, so that it ends up as nearly double by the time you get to 12 years. And the two coloured lines for the Alfred are our donation after circulatory death and our brain death donors, showing that the DCD ones do just as well. So we're very, very proud of those results, and they are world's best practice before, early and late from transplant. So in conclusion, with supporters, friends and a great group of nerds, much has been and will continue to be achieved. And as I say, I slightly got that Friends with Benefits slightly confused. I wasn't really saying that Lucy was emailing Steve about it. It was all properly professional. So thank you to all our supporters as well as team members and collaborators, and a special thanks to the patients who contribute to the research and obviously aiming to help others as well. And sort of in summary, this is the Alfred view of the world, with the Alfred in the centre of up the top there, and our collaborators around, and you can see we have collaborators on many continents, and very pleased to be thinking in the big picture, not just in a small picture. So thank you.